My name is Sophie Kornfeld. The date is October 8th, 2024, and I have chosen to do my presentation on the long and short reflexes of the enteric nervous system. So first we'll start by talking about the enteric nervous system as a whole. The enteric nervous system is a complex network of nerve cells in the digestive tract that controls digestion. The enteric nervous system is part of the autonomic nervous system, which you can see on the slide. And the enteric nervous system is made up of two plexuses, the mesenteric plexus and the submucosal plexus. The submucosal plexus innervates cells in the epithelial layer as well as smooth muscle of the muscularis mucosa. The mesenteric plexus controls and coordinates the motor activity of the muscularis externa. The enteric nervous system can carry out reflexes independent of the central nervous system, but it also can work with it. The enteric nervous system controls motility, secretion, and growth of the digestive tract. Studying the function of the enteric nervous system can be difficult because enteric reflexes have no discrete command center, like the brain for the central nervous system. And then the enteric nervous system, although it's separate from the central nervous system, share a few features. The first feature is intrinsic neurons. Intrinsic neurons of the two nerve plexuses of the digestive tract are those neurons that lie completely within the wall of the gut, just like interneurons are completely in the central nervous system. Autonomic neurons that bring signals from the central nervous system to the digestive system are extrinsic neurons. The second feature is neurotransmitters and neuromodulators. The ENS neurons release more than 30 neurotransmitters and neuromodulators, most of which are identical to molecules found in the brain. These neurotransmitters are sometimes called non-adrenergic or non-cholinergic, to distinguish them from traditional autonomic neurotransmitters like norepinephrine and acetylcholine. Some of the best known GI neurotransmitters and neuromodulators are serotonin, vasoactive intestinal peptide, and nitric oxide. The third feature that is similar is glial support cells. Glial cells of the neurons within the ENS are more similar to astroglia of the brain than the Schwann cells of the peripheral nervous system. Another feature is a diffusion barrier. There are capillaries that surround glia in the ENS that are not very permeable, creating a diffusion barrier, which is similar to the blood-brain barrier of the cerebral blood vessels. Finally, there's an integration center. The reflexes that originate in the GI tract can be integrated and acted on without neural signals leaving the ENS. Neural networks of the ENS is like its own integration center, much like the brain and the spinal cord. So we're going to start with short reflexes. The enteric nerve plexuses in the gut wall act as like a little brain, which allow local reflexes to be begun, integrated, and completely end in the GI tract. Short reflexes are reflexes that originate in the enteric nervous system and are integrated without outside input, so they stay completely within the digestive tract. Short reflexes are usually quick because they're local, which can make it efficient for quick signaling and for changing things quickly. The submucosal plexus contains sensory neurons that receive signals from the lumen of the gut, and then the ENS network integrates this sensory information and initiates a response. Submucosal plexus controls secretion by GI epithelial cells, and the mesenteric plexus neurons in the muscularis externa influence the motility. And so the ability to move things and secrete things quickly is important in digestion and in a changing environment, and so that's where short reflexes would come in handy. And then long reflexes, the ENS can work alone or it can communicate with the central nervous system. It can send sensory information to the central nervous system and receives input from the central nervous system through autonomic neurons. A classic neural reflex begins with a stimulus transmitted along a sensory neuron in the central nervous system, where the stimulus is integrated and acted on. In the digestive system, some classic reflexes originate with sensory receptors in the GI tract, but others originate outside the GI tract. Long reflexes are long reflexes no matter where they originate. It's just important that the digestive reflexes integrate with the central nervous system. So they're still long reflexes even if they start in the digestive tract or they start somewhere else. Long reflexes that originate outside the digestive system include feed-forward reflexes and emotional reflexes. These reflexes are called cephalic reflexes because they originate in the brain instead of in the digestive tract. Feed-forward reflexes begin with stimuli such as sight, smell, sound, or the thought of food, and they prepare the digestive system for food that the brain is anticipating. Emotional reflexes and their influence on the GI tract show a link between the brain and digestive system. GI responses to emotions range from traveler's constipation to psychologically induced vomiting and diarrhea, as well as like the feeling of butterflies in your stomach when you're nervous or when you're talking to someone new that you might like. In long reflexes, the smooth muscle and glands of the GI tract are under autonomic control. In general, the parasympathetic division is excitatory and it enhances GI function, rest and digest. Most parasympathetic neurons of the GI tract are found in the vagus nerve. And the sympathetic neurons usually inhibit GI function. You can see on the left an example of a reflex where that begins in the digestive system and travels along a sensory nerve to the vagus nerve, to the central nervous system, and back to the digestive system. There's just one example. And finally, this is a diagram from the book. And it's just a brief overview. The gray arrows show the pathway of long reflexes, and the red arrows show the pathways of short reflexes. In the center, you see the enteric nervous system in a gray box. You can see that the enteric nervous system integrates many different activities that can lead to different responses. Both long and short reflexes can lead to changes in motility, release of bile and pancreatic secretions, enzyme, acid, and bicarbonate synthesis and release. Just long reflexes, where the brain is the target, can affect hunger. And long reflexes, where the integrating center is the endocrine pancreas, can increase insulin and decrease glucagon. And so overall, this slide shows the stimuluses, the integration center, targets, and their responses. All very similar to what we discussed earlier in the presentation. I think this slide is very comprehensive and has helped me to understand the enteric nervous system as well as short and long reflexes, and I will be using this to help me study. And this is my presentation on the short and long reflexes of the enteric nervous system. Thank you.